So in this lecture, we're going to look at the bacterial diseases that can affect the nervous system. And if you recall, bacteria are the ones that can produce toxins. So most of the infections we're going to talk about are going to be toxin-based. Recall from anatomy that the nervous system is broken up into primarily the central nervous system, which is the brain and spinal cord, and the peripheral nervous system, which involves your spinal nerves that go out and communicate with the rest of the body. Given that the nervous system is such a key organ system, we have lots of different protections. The obvious forms, of course, the skull and the vertebrae to protect the brain and spinal cord. But then we also have CSF, which is the cerebrospinal fluid. It's a blood filtrate that brings in nutrients, removes wastes. We have a series of cells called astrocytes that wrap around the capillaries that serve the brain and form tight junctions so that nothing can leak in between. And this blood-brain barrier excludes most substances. So unless it's really, really small, like oxygen and carbon dioxide, or it's fat soluble, it can slide right through those protective membranes, then it should be excluded from the access to the nervous system. So this is sort of a double-edged sword. By having a blood-brain barrier, we exclude the immune system. So we have to have cells on the inside, which we call microglia. They're basically the phagocytes of the nervous system, because otherwise all the rest of the phagocytes that are in the bloodstream can't get in. This also creates a problem if we want to get drugs into the nervous system, right? They have to be fat soluble to cross the blood-brain barrier, but they need to be water soluble to be circulating in the blood. So it really creates a difficult treatment option. And then recall we have the meninges, which are the layers of connective tissue on the surface of the nervous tissue. So when we have meningitis, it's not the brain that's infected, it's just the connective tissues on the surface of the brain. So we had the outermost was the dura mater, it's the toughest one. The arachnoid mater is the next layer, and it basically has these little fibers that push up off of the surface of the brain and create what we call the subarachnoid space, and that's where that CSF flows. And then we have the pia mater, which is fused to the surface of the brain and basically follows all the contours of the brain. So those three layers are very protective. Now remember, there are these little outpuckerings, what we call the arachnoid villi, that are the exit for that CSF. So the CSF is formed deep in the pockets of the brain, what we call the ventricles, by way of what we call the choroid plexus. So the filtrate comes in through the choroid plexus. The filtrate leaves by way of these arachnoid villi. So you can kind of think of that as a breach in the blood-brain barrier. But because the pressure is essentially moving out, as long as there's enough pressure on the inside pushing out, anything that's in the bloodstream where that CSF is going, which is up there in that gap between the two hemispheres, that's essentially blood where it's going back toward the heart. And so that is a place where microbes could potentially find their way into the nervous system, but that's fairly rare. So again, the central nervous system is the brain and spinal cord. If you divided up the brain into its general neighborhoods, we have the cerebrum, which makes up the bulk of the brain, the superior lobes, and this is where voluntary skeletal muscle contractions occur, are mandated, and this is where we have what we think of as higher thinking, so personality, memory, processing, language, all of that stuff. Cerebellum is sometimes referred to as the little brain. This is the posterior, very convoluted portion of the posterior part of the brain. And this is where those voluntary muscle movements get coordinated. So this is where we do sort of the fine-tuning of when to start and stop contractions. But it's also where a lot of involuntary muscle contractions are managed. And then we have the brainstem, which basically makes up the rest of the brain. And this is usually referred to as the survival center, because this is the subconscious control of everything from heart rate to your vasoconstriction and vasodilation, which controls blood pressure, your breathing rate. This is essentially at the top of the spinal cord. So damage in the brainstem is typically fatal because you can't control those involuntary actions. So the peripheral nervous system then is composed of all the nerves that then exit the spinal cord and transfer those commands either from the central nervous system out to your muscles and glands, or carry information into the central nervous system from your sensory organs and other parts of the body. So cranial nerves are those that extend from the base of the brain, and the spinal nerves then extend from the spinal cord the rest of the way. So there are generally three types of nerves based on their function. So sensory nerves carry information in toward the central nervous system. Motor nerves carry information out from the central nervous system. And mixed nerves would be those spinal nerves as they are making that transition in or out of the spinal cord, where you basically have axons from motor and sensory nerves bundled together. So remember, the action potentials can only travel one way down that individual cell, but we bundle them together in nerves. So you may have areas where you have information going both ways because of the way those cells are bundled. So the main cells of the nervous system that are doing the job of sending action potentials are the neurons. And then we have the neuroglia, which is a whole family of cells that basically support the neurons. So this would be things like those astrocytes that make the blood-brain barrier, like the microglia, which are inside the nervous system, providing that immune function. They're Schwann cells that protect the axons when they're and produce myelin along those cells. So we learned about all those different groups of neurological cells, neuroglia, in A and P. So recall that the nervous system is privileged tissue. That means that because of that blood-brain barrier, large things cannot cross. So of course your white blood cells are some of the largest cells in the body, and that lacks access. So we call that privileged tissue because there's no immune system access. So how do organisms find a portal of entry into the nervous system? They can get in one of these ways. So either you have a break in those physical barriers, like the bones of the skull or the meninges. It could be bypassed altogether by way of invasive or surgical procedures. Or if the meninges, which are the protective lining or covering on those tissues, get infected, then that can create inflammation and create a barrier, a breach in the barrier. Very rarely will organisms find their way into those peripheral nerves and travel the nerve itself to get back to the central nervous system. But the things that can do that would include things like rabies, virus, leprosy, bacteria, or, for example, the brain-eating amoeba. It would use the olfactory nerve to find its way into the brain. So specifically in this lecture, we're going to focus on the bacterial diseases. So typically bacteria cause disease in one of two ways. Either the nervous system cells directly get infected, so for example bacterial meningitis or leprosy, that's where the nerve cells are actually infected, or because bacteria tend to produce toxins, many of the toxins we're going to talk about are neurotoxins. And so recall the term focal infection. You may have an infection somewhere else in the body, but they're releasing toxins that get in the bloodstream and find their way to the nervous system. And so that's what we mean by a focal infection. And so, for example, botulism and tetanus are both examples of that. So again, here's the list from your textbook of all the diseases that can affect the nervous system, with a few extras here in the central box. And so I've also added those additional common names that go with the pathogens that they have listed here. So in this lecture, we're going to focus on the bacterial, but recall your textbook sort of lumps them all anatomically, such as like encephalitis, but there are multiple forms of encephalitis. Meningitis, there are multiple forms of meningitis. So again, your book organizes them slightly different than how we're going to approach them in these lectures. So let's talk about a few of the virulence factors we're going to see through these diseases. Antigenic shift. So this is something we haven't seen quite yet, which is basically the antigens on the surface of any cell are the thing that your immune system recognizes as foreign. So those glycoproteins, which basically identify the antigen, can change. So because they're proteins, if they have the ability to alter the shape of those glycoproteins and shift the shapes, if you will, that means every time the organism replicates, it's going to look different to our immune system, and it might as well never have been seen before. And so you can't develop immunity against organisms that constantly change their shape. Fimbriae, recall these are the little Velcro-like extensions that are on the surface of some bacteria that allow them to adhere to the host tissues that usually we think of as starting the layer of a biofilm, but just being able to attach to any tissue is a virulence factor. Capsules, we've seen again in previous chapters where it's basically a coating on the bacteria that looks like a carbohydrate to the phagocytes, and so it's essentially camouflaged. We're going to meet the mycobacteria in this chapter, which is going to include the leprosy organisms. So remember, mycolic acid is a special waxy component in the cell walls of just the group called mycobacteria. And not only does that waxy component help them from drying out, but it also is going to prevent some antibiotics from reaching the bacteria and being effective. So we'll talk a little bit about that. Endotoxin, we're going to have several gram-negative bacteria, and we've talked about how endotoxin can trigger all of the innate defenses simultaneously and potentially lead to excessive fever and shock. And of course, endospores. So some of the bacterial diseases that we're going to talk about today require deep anaerobic wounds and an endospore getting into those wounds. So think botulism and tetanus. So let's start with bacterial meningitis. There are about 50 different species that can cause bacterial meningitis. We're going to talk about the top 5. But all forms of bacterial meningitis share symptoms, signs and symptoms, which is essentially a sudden fever. So within a few hours of exposure, you're going to have very high fever and inflammation. And so depending on where the inflammation occurs, the symptoms are going to look different. So if it's a cranial nerve, the meninges on a cranial nerve that's inflamed, then that's going to impede CSF flow throughout the brain, and usually results in very severe headaches, vomiting, and pain. Whereas if it's an inflamed spinal meninges, then the pressure on that neuron is going to affect whatever muscle is downstream, right? So for example, if it's high up in the spinal nerves, it might cause stiff neck. If it's lower down, you might have numbness or altered muscle control elsewhere in the body. So it just depends on where the infection starts. So again, one thing that differentiates bacterial meningitis from the other forms we're going to talk about is how quickly it develops. So usually within a few hours, you're going to see an increase in white blood cells in the CSF if you did a spinal tap in any form of bacterial meningitis. We do see that there's a genetic predisposition. So there's something called a toll-like receptor on your immune system cells that basically determine how aggressive the immune response is going to be. So that's why not everybody gets the same type of symptoms associated with whichever bacteria they're exposed to. And if those meninges get infected to the point where the bacteria can get deeper, then it can become encephalitis. So untreated meningitis can become very severe, and depending on what part of the brain gets infected, it could just result in behavioral changes, or it could lead to coma or death if it gets to the brain stem. So again, there are numerous species that can cause bacterial meningitis. We're going to focus on the ones that cause more than 90% of the cases, are these top five bacterial species. So Streptococcus pneumoniae, obviously it's named for what it does in the respiratory system, and we'll see that again later in the semester. But it can also cause meningitis if it gets into the nervous system. So this is the leading cause in adults. This is sometimes referred to as community-based bacterial meningitis because it's out in the working adult population, right? And so this one we do have about a 20% higher mortality rate in adults than other forms of bacterial meningitis. So it is one of the dangerous forms. Neisseria meningitidis is also sometimes considered epidemic, and so this is one of the ones that we vaccinate against when students go to college. They're often recommended to get this vaccine because it can spread through the dorms and other crowded conditions. So this one is famous for having fimbriae in a very thick capsule, and it is a gram-negative. So all three of those are going to be key virulence factors for Neisseria meningitidis. Haemophilus influenza, this actually used to be the leading cause of bacterial meningitis, and primarily in infants, prior to the vaccine that we now give regularly to infants. So this is what we call the HIB, Haemophilus influenza strain B, essentially is where they get that name. So there is the strain A that doesn't cause any disease, but strain B is difficult. So this, like about 95% of cases in babies historically would have this form of meningitis. And because before it was treatable, it would lead to long-term damage. So there was, several generations where if you had deafness, for example, it was most likely caused by this infection during infancy. And so now that we have a vaccine, you rarely hear about this particular version. But also the name reminds you that influenza, so obviously this is not the cause of influenza. Influenza is a viral disease. But when they first were looking for the cause of influenza back in the 1918 pandemic, every patient that they sampled had this bacteria, and that was before we understood viruses. So this bacteria actually got named because every single patient had this bacteria in their lungs, but wasn't actually causing the infection that it got named for. But in science, once the name has been applied to something, it's really hard to go back and change scientific names because there's a lot of research, a lot of publications and things. It's really hard to get people to want to change scientific names, and so this one actually got to keep that name even though it has nothing to do with the influenza. And then there's Listeria monocytogenes, which actually the form of bacterial meningitis caused by this organism is sometimes referred to as listeriosis. And it's primarily a concern in the developing fetus. And so we worry about pregnant women being exposed and immunocompromised individuals. And so the unique virulence factor for this organism is something called listeriolysin. It's essentially secreted by the bacteria, and when it gets engulfed by a host cell, it secretes this substance that basically hijacks the actin molecules that allow a phagocyte to move itself around. And when it hijacks those actin molecules, it uses them sort of as a tail to push themselves into other host cells. So typically phagocytosis is voluntary, right? The cell actually engulfs the organism and brings them in voluntarily. But this allows the cell, the bacteria, to basically force its way into other cells and eventually, essentially evades detection from the immune system because it's inside of the cell. Now, typically we think of viral infections for intracellular parasites, right? Your immune system knows how to go after viruses because they are typically hidden away in cells, but not bacteria. So that's one of its the little tricks up its sleeve, if you will. Then we have Streptococcus agalactae, group B. So this one is still typically if you have a newborn that gets diagnosed with meningitis, it's probably this one, mostly because about a third of healthy females have this bacteria in their vagina. So as a baby comes through the birth canal, it gets exposed and can be infected. And so this is the one they refer to as streptococcal meningitis. So we can lump all those five together as being transmitted through respiratory droplets with a few exceptions. That last one we just talked about, Streptococcus agalactae, that one is not respiratory droplet spread. It is spread through the birth canal. Listeria, also not respiratory, but is transmitted through ingestion of contaminated food. And so the Streptococcus pneumoniae, it is respiratory droplet spread. And because it's in the throat of about 75% of humans without causing any infection, it's readily spread. So again, that Streptococcus pneumoniae is the one that we usually refer to as community-based, and so it can be spread. And when we say not casual contact, right, you typically have to live with the person or spend hours and hours, so maybe a coworker, but not just passing someone in the street with very casual contact. And so meningococcal, if you recall, that's the Neisseria form. That's the only form that really becomes epidemic and is readily spread through respiratory droplets. And again, so crowded places like dorms and barracks, those are the places where we worry about the meningococcal version. Diagnosis is typically based on symptoms, but also to identify which species, they would need a spinal tap and to culture those bacteria. So we do have some vaccines available for strep pneumoniae, so the pneumococcal. We have the HIV vaccine we talked about, and then also for the meningococcal, the Neisseria form. But there's still not a vaccine for the streptococcal form, the Streptococcus agalactae that is transmitted to the newborn. There are lots of different forms of that vaccine they've been working on for decades because of the damage it can do to the newborn. But the forms that are the closest to being approved are vaccines that would be given to the pregnant woman during her second or third trimester. And those forms of vaccines are in their second phase trials right now. And so that's the one that is largely understood to have the best promise for that. But unfortunately, when we say Group B, there are multiple serotypes. So just like anything that has antigen shifts, if it changes its shape every time it replicates, it's hard to make a vaccine because if the shape is different. So in that case, you have multiple serotypes, and that's partly why the vaccine production for that particular group has been so evasive. If you want to avoid listeriosis, avoiding high-risk foods. So think unpasteurized or undercooked deli meats, things like that. Raw milk and raw cheese, definitely not a good idea for pregnant women. But if you're immunocompromised, milks, cheeses, all of those things could be high risk. So bacterial disease, unless it is a resistant strain, we do have effective antibiotics for the bulk of these species. So Hansen's disease is probably more familiar as leprosy, and this is caused by the bacteria Mycobacterium leprae. So recall that prefix myco means fungus. So in this case, this is in fact a bacteria, but when it grows in culture, it looks like a fungal colony, and that's how they got the name Mycobacterium. The virulence factor here primarily is that mycolic acid. So these mycobacterium, they are gram-positive bacteria, and so their cell wall is very thick, and they have this added waxy component called mycolic acid. So diagnostics, pretty much signs and symptoms are fairly diagnostic, but then they can also do what they call an acid-fast stain. And if what they find are these bacilli that can get through the acid-fast. So the acid-fast is basically that mycolic acid makes it very difficult for the dyes of a gram stain to get through that layer, and so you get altered results. So you have to do an acid-fast stain, which requires special acids to dissolve away that mycolic acid. So this is a unique kind of staining, but that if you have a positive result for an acid-fast stain, you're definitely looking at one of the two mycobacterial diseases, which is either leprosy or tuberculosis. And so treatment, just like tuberculosis, is a very long-term antibiotic treatment. Now, partly this, why it has to be long-term is because of that mycolic acid. Those bacteria, when they grow, if you're looking at a typical bacteria like E. coli, they might have a regeneration time of 20 minutes where they can build the next generation. But in mycobacteria, that mycolic acid is so energetically expensive, it may take them 36 to 48 hours to make a new cell. And so if you have a very slow growing bacteria like that, you need antibiotics on board long term for it to have any effect on those organisms. And it's got to be the right one that can actually bypass that mycolic acid. So treatment is typically very long term, and it has to be continuous. Unfortunately, the damage that's associated with leprosy can't be reversed. So even though now we have antibiotics that can treat it, any tissue damage that's done is going to be permanent. So typically we see this manifested in one of two forms. What we call tuberculoid leprosy is very localized, and this one actually kind of shows you the sister disease, tuberculosis. It's the way that our immune system goes to where the bacteria is and basically encircles it with sort of a calcium wall. And that is what a tubercle is. So when we get to tuberculosis and we talk about how you can confirm it with an x-ray, you're basically going to see those calcium-rich, walled-off little round areas called tubercles. And so this one, when we say non-progressive, meaning it's not going to migrate, it's not going to spread further than those tissues. However, if the immune system is actually capable of getting to where those bacteria are, it can cause progressive tissue damage, and this is what we call lepromatous. And this is typically what people think of when they think of leprosy, is the tissue loss because of the extensive tissue cytotoxic T cell. So this is another one of those where your immune system really is the thing, it's sort of self-inflicted damage. So this is a disease of Schwann cells. So recall that these are the cells that wrap and protect the axons. So if your axons are no longer healthy, they can't send action potentials, and those tissues will start out with a loss of sensation. And then wherever those virally infected cells are, the cytotoxic T cells will come in and trigger apoptosis, which is then what causes the loss of those tissues. So we tend to see the damage associated with this bacteria in the extremities. So think your digits, anywhere on the surface of the body, or the face, the ears, the nose, things like that, were usually lost first because those were the cooler parts of the body, and that's where that bacteria tends to grow best. And so it turns out that armadillos, which are a mammal that actually has a fairly low core body temperature, are a really good reservoir for this bacteria. So the mode of transmission is actually still not 100% understood, but we presume it to be person-to-person contact by way of prolonged contact, right? So think caretakers of an individual that is infected. And so we presume there's probably a break in the skin and or respiratory droplets, but again, it's not casual contact. It's probably caretakers or living in the same home together. We also know that there have been cases tracked to handling and eating armadillo, and especially in parts of the world where they eat raw liver from the armadillo as a delicacy. So places like Brazil, it's actually now illegal to eat raw liver because of the risk of transmitting this bacteria. So While we don't vaccinate against tuberculosis all over the world, there is some evidence that because of the similarity between the two species, the BCG vaccine, which is against tuberculosis, may actually confer some protection. But it turns out that the genetic bottleneck that occurred back when the plague essentially killed a third of humans on the planet, that was also a time where leprosy was very common. And so it turns out that the gene that basically got weeded out was the susceptibility to leprosy. So it turns out, genetically speaking, about 90% of humans walking the earth today are naturally immune to this bacteria. Where you still see susceptibility to this bacteria tends to be in those places of the world where the plague was not very common. And so we're talking about, the South Pacific Islands, places like the Philippines or Micronesia. And so you see basically those people with that lineage are the most likely to be exposed and get infected from this bacteria. And we also don't see this disease very frequently because now we have antibiotics, so it's unlikely to progress to the visible form that we think of. We still have certain parts of the world, so again, in Brazil, they still see some cases, but again, it's treatable, and so it's not nearly the stigma, and it's not nearly as common as it used to be. So these are the places in the world where we actually still see some cases. And again, sometimes it's been tied to the exposure to armadillos. And it turns out the armadillo just are reservoirs, right? They don't actually suffer the disease, mostly because they are not very long-lived creatures. And so they don't live long enough to actually get the disease themselves. So then there's the toxin-based diseases. So for example, botulism, right? So Clostridium botulinum, recall that Clostridium and Bacillus are two bacterial species that are common in the soil worldwide. They make endospores. And so this is, when we think of botulism, we tend to think of food, and typically we think of home canned foods, right? So if you can your vegetables, but you don't meet the requirements of time and temperature and pressure, then those soil, which contains the endospores, might still be on those vegetables, and then you ingest the toxins that were produced in the can while it was in that anaerobic space. So primarily beans, corns, and peas, primarily because of the pH associated with those particular vegetables. But when we say botulinum toxin, there's actually 7 different types, and each has a slightly different virulence, if you will. But these are neurotoxins, and these will permanently bind to the axon terminals of your motor neurons. And so the toxin can be produced, whether it's produced in the can and then you ingest the toxin. We'll talk about the different forms of transmission. But basically, no matter how the toxin is formed, it will circulate the body in the bloodstream and then find its way into that synaptic cleft and will permanently bind to that axon terminal to where your motor neurotransmitters can no longer be released. So by default, the muscle downstream from that can no longer receive messages, and it basically paralyzes that muscle. So there are 3 primary forms, and it's sort of all based on how you're exposed. So foodborne botulism is just what it sounds like. You're ingesting some food that contains the pre-made toxin. So for example, again, you have endospores get into a can that wasn't properly treated, so those endospores didn't get killed. The organism wakes up, starts metabolizing and producing toxin. And so when you eat whatever was in that can, you're going to ingest a certain amount of the toxin pre-made. So within a very quick period, within a day or two, those muscles, as the toxin goes and binds to and blocks that acetylcholine release, you'll start to have paralysis of those muscles. Now, if you ingest just a small amount, then it'll only affect so many muscles around the body. But if you ingest a large amount and that toxin finds its way, for example, to the phrenic nerve, which controls the diaphragm, then you could suffocate before those nerves have a chance to regrow a connection with that muscle, because that can take months for those axon terminals to replace themselves, right? So as long as the rest of the nerve is healthy, your nervous system is very plastic, meaning it will intentionally build new nerve endings to grow, to recommunicate with whatever it was originally communicating with. Infant botulism is where the endospore is ingested. And so typically we think in infants, things, again, improperly canned foods, in this case, now they can ingest pre-made toxin, but that would be foodborne. Infant botulism is when they ingest the endospores in places like raw honey, right? Think about, why would honey have endospores? Because any soil containing endospores that gets raised in the dust, or in the wind, gets stuck in the honey and then if it's ingested without being pasteurized, then you're ingesting endospores. And because in an infant, you don't yet have a mature immune system, those bacteria could actually end up in the gut and start growing, and then the toxin is being produced in the baby's body, as opposed to foodborne, where you're ingesting the pre-made toxin. Now, of course, in adults, we have mature immune systems, and typically endospores can't grow in the GI, and so that's why this typically is associated with infants as opposed to adults. And so in babies, we're going to call this, basically, there are some very non-specific symptoms. So muscle weakness basically just reflects that paralysis that's happening. If you had a baby that maybe was fairly vigorous, it would cry loudly when it needed something, but then it starts to become sort of a weak whimpering cry as opposed to a full throated cry. It's not very hungry. It doesn't hold its head up as it used to, so we would generally refer to all of that as failure to thrive. Can be a sign of botulism. And very oftentimes, because the toxin is actually being produced in the GI and then being absorbed and moved around the body, one of the first signs is constipation, which of course in an infant it's kind of hard to tell. They're either going or not. And so constipation might not be the obvious sign. But again, if that progresses to what they call floppy baby syndrome. So basically, if you have a baby that used to be able to hold its head up and then all of a sudden is no longer able to do that, it could be a sign of infant botulism. And then there's wound botulism. So this one's similar to tetanus in the case that if you get endospores into a deep wound and the bacteria then starts to grow in that anaerobic space, releases toxins, and then can go around the body and mimic foodborne botulism. So because these toxins are so good at paralyzing muscles, we actually use them both clinically and cosmetically. So when we think of Botox injections, right? So facial wrinkles, if you can keep the skin from wrinkling because of the muscles underneath, then you can reduce the production of long-term wrinkles. Therapeutically or clinically, there are forms of botulism that are used for chronic migraine. So that's actually usually targeting the same muscles of the face. So if people tend to carry tension in their face and they're going to have potential migraines. In some cases, so eye movements, eyelid movements, what they call spasmodic torticollis, where you have neck and head spasms, those have been treated effectively with botulism. In some cases, things like excessive sweating and over saliva production have been treated with botulism. Even cases of overactive bladder, if you could paralyze those muscles that are hyperactive. And even some weight loss treatments. So if you can keep your stomach from growling all the time, then maybe you can avoid that hunger that causes weight gain. And so some obesity treatments now are even using botulism. And so they literally inject botulism toxin into the muscle of the stomach and intragastric injections. So prevention really depends on which form you're talking about, but proper canning is the primary form of avoiding botulism. And so that's why we have very standard industrial regulations for food that is sold to the general public, right? Because they have to meet a certain time and temperature and pressure to destroy those endospores. Proper wound care can avoid that wound botulism. Most wound botulism is actually associated with IV drug use. So using dirty needles can lead to botulism more so than anything else. In very young babies, so under 1 year of age, we tend to avoid feeding honey, especially raw honey. So that's why in most grocery stores now you see it's actually fairly hard to get raw honey commercially, because we now pasteurize honey to avoid this problem right here. But it's better to be safe than sorry and avoid feeding honey until their immune system is more mature. In agriculture, we have a variety of things that can cause what are known as downer cattle. So there are certain fungal diseases and other things, but botulism is not uncommon in decaying feed. And so that's another thing that can be affected there. And so how do they identify botulism in cattle? They would have excessive drooling, and when we say loss of apprehension, right, most animals are a little bit startled when a human comes near them, but total loss of apprehension and excessive drooling are two good signs of botulism in cattle. And again, why they call them downer cattle, if that toxin then finds its way to the diaphragm, they can no longer breathe. And so typically it's respiratory failure or actually suffocation that kills those animals. So treatment really, again, depends on the form that you are exposed to. If it's foodborne, you've only ingested the toxin, so really there's no way to remove the toxin. But if it's infant botulism, they can do gastric lavage and remove the bacteria from the intestinal tract. We do have antibodies that can be used that are going to target those toxins. So again, we don't target the bacteria because the bacteria is really not doing the problem. The problem is the toxin. If you can keep that toxin from permanently binding to the neurons, then you can avoid all of the issues associated with the disease. And of course, it is a bacterial infection. So if it's especially infant botulism or wound botulism, where you actually have a growing bacteria in the body, then antibiotics are effective. But again, because you're just ingesting pre-made toxin for foodborne, antibiotics aren't going to help in that case. And of course, and we don't have a vaccine against botulism. Antibodies are different than vaccines. Recall that treating after the fact with an antibody is very different than preventing ahead of time with a vaccine that triggers your body to make those antibodies. So botulism is fairly rare in the U.S., and most of those cases are going to be wound and foodborne botulism. And of course, there is a fairly high 10% mortality rate, even though we know how to identify it and we have treatments. But infant botulism is still the most common form, and it is, again, typically associated with honey consumption, which is why we pasteurize most honey now. In Europe, just this year, about 87 cases were documented after those intragastric injections for obesity treatments. So this is not a very commonly used treatment for obesity, and now we're seeing there that may not actually be the risk benefit there may not be. And then tetanus, this is a sister species to botulism, so Clostridium tetani, also an endospore-forming bacillus, and required to get those endospores deep into an anaerobic space like a wound in order to germinate and grow. So this one tends to have a fairly long incubation period. So it can be weeks from when you had exposure to the endospore soil getting into the wound and when the bacteria actually start growing. So botulinum toxin, if you recall, removed the ability to stimulate a muscle. Tetnospasmin toxin works a little bit differently. It also will permanently bind to nerves, but in this case, it has an affinity for the inhibitory neurotransmitter side of things. So we don't really talk about it much, but when we talk about how to stimulate voluntary muscles, remember you have antagonistic muscle pairs. So for example, the biceps on the anterior surface of the arm and the triceps brachii on the back or the posterior surface of the arm, those are antagonistic muscles. They do exact opposite jobs at the elbow joint. And so we always talk about the voluntary activation of the biceps, for example, when you want to flex the elbow. But what we don't really talk about is the fact that your involuntary subconscious control of your body is sending an inhibitory message at the very same time to the triceps, because you can't actually contract both of those at the same time, or else the joint you wouldn't get any movement at the joint. And so what tetanus does is the tetnospasmin toxin will go to the opposite muscle, the antagonistic muscle, and block the inhibitory muscle, and so or message. And so eventually every inhibitory message that gets inhibited, you're going to get muscle contraction, but no inhibition. And so you get tetany, right? And that's what tetanus means. It's basically a contraction that won't let go, is tetany. And so signs and symptoms, this is also sometimes called lockjaw, because one of the first symptoms typically we see is those muscles of the jaws and neck will contract and not release. So if they can't inhibit those muscles after a contraction is over, then that's a problem. And so basically the lockjaw, you can see in the picture on the left here what we call the sardonic smile, right? So those muscles, the teeth clench and stay in that position. You'll see some fever and muscle spasms throughout the body, but again, just like botulism, if that permanent binding of the toxin gets to the diaphragm or the nerves controlling the diaphragm, then it can no longer relax and you can no longer inhale and exhale properly. And so that is going to lead to respiratory failure. And so you can also see in these images, it's the stronger of the two antagonistic muscle pairs that typically wins, right? And so you can see all the fingers and the wrist and the elbow are flexed and pulled close to the body. And the baby on the right side, your back muscles are much stronger than your abdominal muscles, and so you get that arched back. And so that tetany that you're seeing is basically the stronger of the pairs of muscles is usually going to win out. So of course the most susceptible are unimmunized individuals. We do have a vaccine that can prevent this infection. Circumcision patients. So in other parts of the world where we don't have regular vaccination against tetanus, it's unfortunate that, not only do you potentially just have a dirty wound, but in some cultures they actually do ceremonial fires, and they will take some of the ash from those ceremonial fires and rub them on the circumcision wound or on the umbilical stump wound as part of their cultural rituals. Part of that is to help clotting and cause it to stop bleeding. But because those fires are burned on the ground, there's some dirt in those fires, and so that's typically how it's getting transmitted to the patient. And again, also for umbilical stumps, and it's really just a cultural norm in some parts of the world, but that's typically where it gets into very young patients. So prevention, of course, we know there's a very effective toxoid vaccine. And again, this vaccine wasn't made against the bacteria, it was made against the toxin because it permanently binds to those nerve cells. So if you get exposed to the bacteria but it's not actually producing the toxin, it's not going to do you any damage. So it's the toxin we need to bind up and neutralize before it can do the damage. And of course, we know that it requires a booster because toxoid vaccines, because it's not made against an organism that can replicate and really trigger a strong immune response, we typically have to give boosters for those types of vaccines. So we know in this country we usually do a regular 10-year booster, but if you have some sort of exposure, right? So the old wives' tale is it's a step on a rusty nail, you get tetanus. Well, it's not the rusty nail. It's the rust suggests that that nail has been outside and has been oxidized, and it's been out there long enough to get dirty, and the rust provides a place for dirt to sit, to be attached to that nail. The nail being it's a puncture risk, obviously, and so the dirt entering the wound is what causes tetanus, but the exposure is anything that causes a deep wound. And so it can be a barbed wire fence, it can be a snake bite, right? A snake that lives on the ground and eats animals that might have had dirt on their feet. Will potentially lead to a tetanus shot because if you have some sort of known potential exposure and you've been maybe 5 years or 6 years out from your last booster, it's better to be safe than sorry and go ahead and give that booster shot so that those toxins don't go and do their permanent damage in your body. So treatment starts with cleaning the wound to remove any additional endospores. Antibiotics, of course, historically penicillin was effective, but now today we would use other forms. And we do also have antibodies against the tetanus toxin. And so that would be something we would do after an exposure to help make sure and bind up and neutralize those toxins. But just like in botulism, depending on how much toxin is in the body, it may take a few months to grow new axon terminals to regain access or regain function of those muscles. But if that tetanus toxin has affected the function of the diaphragm, then that patient might need a respirator to get over the hump of waiting for those axon terminals to grow. So again, it's mostly a problem of other countries where they don't have regular routine vaccination.